Welcome back to our podcast. So our first episode where Dora and I talked about million-dollar decision-making for cleanup sites. And today we have Dr. Frank Loeffler with the Department of Civil and Environmental Engineering at the University of Tennessee, distinguished professor, well-known researcher, Goodrich Chair of Excellence in Civil Engineering, many awards that Frank has won over the years, but very glad to have you, Frank.
Thank you very much for having me. Excited to be here.
I'm Dr. Sam Rosolina, Vice President of Applied Innovation here at Microbial Insights. And so we wanted to just have a conversation around what multi-million-dollar cleanup looks like in the real world and what that means to you, especially in the context of microbial analysis. So when you hear that phrase, multi-million-dollar cleanup decision, what role do you feel like molecular biology or microbiology as a whole plays in that?
So when I hear these large sums of money, right, being used for cleanup, I'm a little worried that people invest resources in a fancy technology that does not rely on microbiology. But cleanup, happens in soils, in sediments, in subsurface environments. And all of those environments are the home for many microbes. So no matter what fancy technology you implement, the microbiology will be there. And it will either interfere with your fancy technology or it will support contaminant degradation based on the more expensive technology. And I think the key here is to understand what is the relative contribution of the microbiology to contaminant degradation in addition to the fancy technology you're implementing. And if you consider the microbiological component in this system, maybe you could reduce the investment up front, in this more expensive, fancy technology, because the microbiology will contribute most likely to contaminant destruction. So this was my first thought when you said, a multi-million-dollar investment. If people designing this system include the microbiology in their project, right? And they count on microbiology as a component to contaminant degradation, that's fine. But I've often seen these projects, to be focused on fancy technologies, ignoring the basic fact that microbiology will always be there.
Right.
The other thought I have that comes to mind, you spend a lot of money on cleaning things up, and mostly for technology. And my worry is that, do we have the people in place that can actually use this money efficiently and in a meaningful way to implement this multi-million-dollar investment?
Right.
And so this, in my mind, requires trained personnel that is available to implement the fancy technology, understand the microbiology, and bring this all together.
Yeah, that's interesting because Dora and I, in our previous episode, we were talking about the importance of having not just experts, but the right experts, right? I think in a lot of these scenarios, there are consultants or engineers who have a solid understanding of remediation broadly, but not necessarily some of the clear understanding of the microbiology, for example, or how it slots into some of these, what you call fancy technologies or other strategies. So I think that's a really good point. Now, can you kind of give some examples of what you're describing as fancy technologies, or are these like novel treatment strategies or?
So I'm a microbiologist by training, so I'm more focused on microbial processes and how they can contribute to contaminant degradation.
Right.
But then over the years, I've also been exposed to other technologies. And an important realization was that no single technology can probably achieve the best possible goals in terms of contaminant destruction when applied in isolation.
Exactly.
So the idea is, it's very clear, at many places that are contaminated, it's probably a good approach to combine technologies, right? So you have some chemical physical treatment, what I call fancy technology.
Yeah.
But the idea would be you take a fancy technology, a chemical treatment, a physical chemical treatment, and integrate this with microbiology.
Yeah.
Right. And then some people also call this a treatment train approach. So you first do something physical chemical. You will remove a lot of contaminant mass, but not everything.
Right.
And then you have the microbiology, taking care of the residual contamination, for example. But even if you don't do it in series, right, even if you implement, let's say, a thermal technology, right, you heat something up,
Right.
the microbiology will be there. Maybe it's too hot in certain locations, but in other locations, you bring the temperature, up a little bit, and microbes like it. Now it's their perfect temperature to make a living, and you actually accelerate contaminant degradation rates. So the point again is when you take a physical chemical treatment, the microbiology will always be there.
Right.
And you have to understand that and know about it so you can take advantage of the microbiology and combine it with your physical chemical treatment technology.
Right. Exactly.
And I think this is also important in coming back to the workforce, right? You have microbiologists, you have chemists, you have hydrologists, you have environmental engineers, and they need to be able to communicate and work with each other productively.
Right.
I've been in this situation many years ago, right? Being a scientist, and then I was thrown into the cold water, was forced to work with engineers, and we speak in our science, in our research, we use different terminology. We have different words. We communicate a little differently. So it's very easy for an engineer to, when I say something, they would maybe say he's arrogant. But I feel the same way when the engineers speak to me, right? Because we don't fully understand the terminology that's common in their field. And so to overcome these hurdles, right, you have to make an effort.
Right.
And it becomes easier when a student is more broadly educated. So an engineering student has already heard these terms that engineers use all the time. And the engineers have already heard something about microbiology and chemistry, some buzzwords. And then it's easier for them to build teams, collaborate, and then they can more easily bring these multi-million-dollar remediation efforts, right, to fruition when they integrate their knowledge and apply this at field scale.
Right. And so that is kind of a hope for the future. I'm curious in terms of the current situation, and there are kind of lots of different groups that we want to communicate with as scientists, but also as microbiologists, as Chen experts. There's, groups like, and Dora and I were talking about this previously, there are groups like the community, and we'll talk about East Palestine, Ohio a little bit. But in terms of, translating to that community, why were we there? What were we doing? But there's also other groups that may not have this scientific or technical knowledge, like the decision makers within companies. What do you suggest in ways to make that communication more accessible?
These are numerous very important points. So, of course, companies that implement remedial technologies, most companies have their pet technology, right? That they have a lot of expertise and they have done it successfully in many different places at different sites. So for them to change to a different technology, there's a delta G, a hurdle to do this, right? And so they may not always be open-minded to what is really the most efficient, cost-efficient technology for a given problem at a site. They rather go there and they want to implement their technology.
Right.
And this is difficult to change, right? Because most companies are specialized, right? But when you talk about a multi-million-dollar, a large investment, right? I would expect consulting companies, whoever's in charge, to be open-minded, look at the available technologies and have the right experts on the team that makes decisions, and then maybe splits this up. No, it's not one technology. There are multiple technologies that come together. But this is not easy to overcome. I don't believe I know any company that is equally strong in all technologies, right? Most companies have their strengths.
Exactly.
Right. And so then this becomes a little challenging. But one point, let me make this clear again. No matter what technology you're implementing, the
Microbes are always
microbiology
there.
will always be there.
That's right. But not always the right microbes, right? Which is why it's important to do some
Correct.
due diligence.
So you have to, if you run a count on the microbial component in contaminant degradation, you have to have information about the microbiology. Microbiomes are different in different areas, in different environments, and at different contaminated sites. So you have to sufficiently characterize the microbiology, the microbiomes, and monitor them over time. Only then can you meaningfully integrate this with your physical-chemical treatment. And now you talked about regulating community, right? And so for them, of course, it's also challenging, right? They may have gotten an advanced degree, right? And they have worked in remediation with a certain technology. But again, now they have to make choices and they have to know about all technologies, the entire breadth of technologies that is available. And these technologies are advancing. This is not a static pool of technologies, right? We add more options, different types of technologies. The regulators have to constantly learn, right? And expand their horizon about the different technologies. If they know enough about the available technologies, right, then they can probably make an educated decision on what technologies may be most appropriate for a given contaminated site. And so here I also take the academic community, right, including myself, right? The academics have to reach out and educate. Because in academia, that's where often the new technologies are developed. We have to inform the regulating community. Now, I have colleagues, they would argue, well, we publish papers, they can read this. Yeah, sure. But this is maybe a little naive, right? So you have to go to meetings and they'll reach out to you. The regulators invite you to give a talk, right? You have to be very open-minded and do this because this is very important. Otherwise, the gap between the cutting-edge science and the regulating community is widening.
Right.
And I'm not talking down on regulators, to be clear, right? I think this is a very demanding job. And staying abreast of all the new technologies, right?
It's very difficult. There's a lot coming out.
I'm sure they try very hard. I think the academic community and maybe also the consulting community, right? We have to educate them to make sure they know about all the newest technologies.
Right, yeah.
And so then, you have a very well-educated community at the regulatory level, at the corporate level, right? The companies, and you have a workforce, and then things look good.
Yeah, absolutely. So a couple questions related to that. One is, do you feel that, because you've worked at that for a long time, and I've heard you talk about being one to be proactive in helping regulators understand what's new and what's moving forward. One of the things that I admire about that is that you are academic, but you're not solely academic. You're not someone who stops at once the work is done in the lab, because you do want to see it enacted in the field, which I greatly respect. I'm curious if you feel like that education or help to be proactively teaching has paid off, and that you're seeing microbiology and bioremediation more and more accepted and more common in a lot of these strategies.
Yeah, absolutely. So I consider myself an academic, but maybe now, looking back at a couple decades of doing this, maybe I'm not the typical academic, because one of my major motivations always was do fundamental research, work in a laboratory, discover new things. But I also wanted to make sure that it has an impact on the environment, a positive impact. The way it worked out, I was interested in chlorinated contaminants and how they are degraded. And then I got in touch with bioremediation. I learned a little bit about this. And then this was in the mid-90s, had the opportunity to explore anaerobic microbes, how they can reductively dechlorinate chlorinated solvents. And this was at the time a new field, and it worked nicely in the laboratory. And then I was a little surprised how strong the pull was from the applied side. Oh, you have a new process. Come to me. We need to test this in the field.
Right.
And this was a little surprising because what I experienced before I worked on reductive dechlorination, there was not so much interest in using microbes for bioremediation. This reductive dechlorination really made a big difference. I now understand that better. It's mainly because these chlorinated solvents were a big problem. Technologies were no good technologies existed. It was all expensive and had to run for decades to centuries, right? So it was all kind of not very productive. And so the community was really ready for a new technology. And then these microbes that can do reductive dechlorination, basically detoxify tetrachloroethene to non-toxic ethene. This was a new thing and people were excited about it. And then, I was forced to work with the engineers. They were forced to work with me. There was enough interest on both sides to overcome these hurdles. And then things worked out beautifully. This is how I started doing all this.
I think a lot of that comes down to making sure that it does seem to be much more accepted since...
Okay, so the academic thing, right? So certainly we publish papers. This is kind of the bread and butter what we have to do also to make our students successful. So we publish and we get grants, and we do all this. But I was always interested reaching out to people, explaining how this approach, this technology, this process can work in the environment. So I was always motivated to talk to regulators, to talk to practitioners, right? And I always attended meetings were mostly attended by practitioners, not so many academics. And I enjoyed being there and communicating with them. And, maybe you can say educating them, but I didn't go there to educate them. I went there to talk to them, and
learn yourself.
And learn myself, right? And make contacts, get samples from them, come up with new ideas. And this advanced my science, my group's science. And it also had the practitioners, right, to better understand the requirement for the process. And then they did a great job, right, advancing this. And now this for chlorinated solvents is a robust technology. Microbial Insights is a key player here doing the monitoring, right, for many years. And I explained to you that this is very important. This has paid off very well for me and I think for this bioremediation practice.
Yeah, absolutely. So you've kind of talked about how it is starting to be more commonly used. You also mentioned that it's important to use in conjunction with all of these other technologies. How would you say, or what would you say to someone to explain to them the risk involved in the uncertainty or not doing the microbial analysis at a site versus the gain involved in especially upfront analysis of what microbes are there and then continued monitoring no matter what the strategy, as you said?
So the uncertainty comes from the fact that monitoring is not done.
Exactly, right.
Okay, so it's very clear the tools exist, right? So we have very good tools that different academic laboratories and companies have designed to monitor microbes that are involved in certain contaminant degradation processes, for example, chlorinated solvents, right? So this is a very robust suite of tools that can be used to assess if these types of microbes with the right genes to detoxify these contaminants are present. These tools can also be used to demonstrate that these organisms, when you implement a bioremediation scenario, right, that they are growing. So they're increasing in abundance over time. And this is a clear indication that the process is working. So these tools are essential to get this type of information. So not applying them, this makes no sense to me. Really, you have to do this. Otherwise, you're fishing in the dark.
Right.
It's a black box. And so this black box, I'm in the civil and environmental engineering department, and this is always the perfect example, right? Civil and environmental engineers, what do? They do wastewater treatment.
Right.
So that's a traditional civil and environmental engineering discipline. And so the wastewater treatment plant works fine most of the time. But then if something went wrong or does go wrong, the engineers don't really know what to do, because the microbiology that runs a wastewater treatment plant has been a black box for them for a long time. And now that these molecular tools are available, right, they became available many years ago for wastewater treatment. So now they understand what these tools can do for them. And they realize what they need to change to make the wastewater treatment plant operate properly again.
Right.
So opening this black box using molecular tools was critical to make wastewater treatment much more robust, run it, cost efficiently.
Fewer emergencies,
and they know immediately how to respond. And this is the same situation, right, in a subsurface environment. If you don't see and you consider this as a black box, it may work, but it may not work, and you don't know. So then you add electron donor and do all the installations, but nothing works because you don't understand the microbiology.
Right.
So if you have the opportunity to learn more about the microbiology, you ought to use these tools. That makes sense to me.
Absolutely.
Yeah.
So taking that information and kind of turning it back towards East Palestine, Ohio, which we had a study together in. It was a big, high-profile site. It was definitely a multi-million-dollar cleanup site. We know that mistakes were made, and so kind of looking to the future, were something like that to happen again, what type of upfront analyses would you think would benefit that type of situation before some of these more drastic decisions were made? Granted, of course, they're made under a lot of pressure and time constraints, but there are certain things that I think we know could have been done a little better. So what analyses or removing that black box would have helped in that type of situation?
So East Palestine was somewhat a unique situation. So we studied the degradation of chlorinated solvents, tetrachloroethene, trichloroethene, for a long time. And one of the intermediates of reductive dechlorination is vinyl chloride. And this is very toxic. It's a human carcinogen. And so we studied how can this be further dechlorinated to non-toxic ethene in subsurface environments in the context of chlorinated solvent remediation in subsurface environments. So now, of course, vinyl chloride is also the precursor to make polyvinyl chloride, PVC pipe.
Right.
And so vinyl chloride is produced in the southern part of the United States, and the PVC is typically produced in factories that are in the northern part, right? And so this vinyl chloride has to be shipped on trains. And the accident there was that several cars carrying vinyl chloride derailed and the vinyl chloride was released.
Right.
If vinyl chloride would not have been an intermediate in chlorinated solvent degradation, people would not have really studied.
Right.
We would not know about its biodegradation so much.
Yeah.
We would not have had the knowledge, how the biomarkers, we would not have known how to do all this.
Yeah.
So my key point here is that funding research, educating people, and then suddenly use this knowledge in a different context was critical for us to go there and do our analyses and demonstrate that these organisms are ubiquitous in this environment. They attenuate vinyl chloride, and it's very unlikely that this is a long-lasting contaminant in this system. But if we would had not had this expertise, right, coming from the chlorinated solvent remediation degradation studies that we built over many years of research, we would not have been in a position to go there and efficiently help.
Right. Yeah.
People argue that fundamental science is useless. Only do applied science. Okay, so we did applied science, but in a different context, and suddenly it becomes very important for this accident. Another example is the laser was invented as a fundamental science project, more than 100 years ago, and we didn't know what to do with a laser.
Right.
So for many decades, this was completely useless.
Yeah.
But now this is super relevant for surgery and all kinds of other things. I think I make a pitch here for fundamental research. This is important, right? Because it will help applied science tremendously in the
Right.
future, even if you can't recognize the application at the moment of discovery.
Absolutely.
So I personally like, linking fundamental research with an application right away, but there's also, a justification for purely a fundamental research,
Sure.
and the application may not be immediately
No,
visible,
that's
known.
right. Yeah.
And this to me is an example for this, because we did applied research in a different area,
Mm.
which suddenly was super important for this type of accident.
Right. And I would say that Microbial Insights is an example of a group that can sometimes take that fundamental research and take it that next step, even if the academic labs are not.
I totally agree. So, and over the years, I talked to Microbial Insights and other companies, go to these meetings where mostly practitioners are, and they tell me what their needs are.
Mm.
And then I realize, hey, this fits my fundamental research ideas, right? So, and I can maybe adjust a little bit. So I personally appreciate this very much because it connects me better to what are the needs, right, to make remediation more successful or other technologies. I think this is important. If you are just stuck in your fundamental research community, you may do some cool experiments, but they may not be important for applied research. But you can actually do very good fundamental research if you talk more often to practitioners who tell you, this is important. And then I always realize, hey, this is exactly what I can do in the laboratory. This is
Here's where I can make the
fundamentally super important, and it has relevance for the application.
So it feels like, kind of wrapping it all in a nice, neat package, that the theme for this conversation was information flow. Because if we're talking about multi-million-dollar decision making, it's ensuring that the right decision makers have the information, whether it's the data, whether it is understanding which analyses to do in the first place and when to do those. It's also information flow to different groups like business people to community people. It's also information flow between engineers and scientists and microbiologists. So a lot of really key parts of that would be some of that information communication and ensuring that people are understanding the value of some of these tools to save them lots of headache and potentially lots of money down the road.
Yeah, of course. I mean, when you implement a remedial technology, right, you don't want to spend more money than you have to.
Right.
Right. I mean, I think this is very clear. And so you look for the most cost-efficient solution for your problem. And in order to do this, you have to have a comprehensive picture understanding of the technologies available. And you have to take full advantage of all the monitoring tools that are available.
Right.
This black box approach, if you treat it as a black box, maybe you're lucky, and it works, and you did not spend much money on monitoring tools, and the results are promising. I would call this a lot of luck. Most of the time, you need to respond to something.
Right.
And if something doesn't work out quite right, you need to know what the problem is or what the problems are. So then you need data. And the data are from monitoring, right? The microbiology with different molecular biological tools, with geochemical measurements and other things, stable isotopes. There are many different monitoring tools. And then you can put the data together and you can respond and potentially save a lot of money that was already invested, right? And if you would not be able to respond properly, it would go down the drain. Right. So to me, this monitoring, looking into this black box is crucial.
Absolutely.
Otherwise, this is too much risk. And you also need to have the data in the end to convince the regulators and the general public, right, based on data that your remedy was actually successful.
Yeah.
Right. So just saying, oh, the contaminant can no longer be measured, it must be degraded. This is probably not enough, right?
Yeah. Nor should it be, right?
So, this thought is that you have to have a multiple lines of evidence approach that remediation is working. And the multiple lines of evidence approach is the contaminant is disappearing. you may find products of contaminant degradation, and you can link it to geochemical measurements, but also to the microbiology. Right. And you can obviously then also talk to the public, right? Much more convincing. You can say, we have this evidence, this evidence, this evidence, and you can assure them that the environment is safe again.
Yeah, absolutely. Well, this is great. Thanks for this conversation. Any other last thoughts on big decision making for especially multi-million-dollar cleanup?
Yeah, so when you make big decisions, you need big people who can make these decisions, right? And people who can make these decisions must be well educated.
Yeah.
So to me, cutting back in education is the wrong approach,
Absolutely.
because you will spend a lot of money on something that will ultimately not work. So I'm worried that big decisions are made without having the educated workforce and the right decision makers in place in the future. We are still in good shape right now, but I can tell you from my experience as an academic, attracting graduate students, attracting students in general is becoming more and more difficult. It is becoming more and more difficult to get external funding to do research. And without a doubt, that translates ultimately to a workforce that is not at the same level in the future as it is right now. Right? And my thought is that we improve, right? We improve over time. But this will become increasingly challenging, because I think we'll go, maybe we'll be stagnant. That would be probably a success, but I'm afraid we go in the wrong direction. If we don't have these people, then everything will become much more challenging.
Absolutely. So that's kind of a reminder that this is an investment as individuals and as a community, as a country, ensuring long-term health, right? Because you're lucky enough to or privileged enough to see that trend over time. So you get to see what's kind of coming down the road in the future based on...
I know people who look at contaminants as problems from the past, right? This happened in the past, something got contaminated, and now we have to deal with it. Politicians, and people in general like to spend money directed in the other direction, into the future, right? Not dealing with past stuff, so you want to do something in the future. And so my strong argument is we will always have contamination. You have to educate the workforce today to deal with the problems of the future. And there's no doubt in my mind, we're doing right now things that we think is okay, but they cause environmental harm, maybe even human health concern. And we haven't recognized them yet. We will recognize them in 10 years, for example, right? You have to have this workforce in place to deal with this problem. If we don't educate them now, if we don't build this workforce, we will run into big problems in the near future. This is my concern. And that's maybe my pitch to everybody.
No, that's a great...
Keep pushing in this direction. It will be very beneficial for everybody.
Yeah. Good call to action.
Yeah.
And get involved, right? Help people
Get
understand
involved,
the importance,
and
communicate the science.
Yeah, and encourage students, right, to seek higher degrees at universities, and talk to politicians that the funding for this type of education is very beneficial for societies.
Right. Yeah, an investment.
Yeah,
Right.
and business, right? This is a business.
Right, yeah.
And it helps the environment, it helps human health, it helps everybody.
The economy, right?
Yeah, absolutely.
Absolutely. Thanks, Frank.
You're very welcome.
It's always good to talk to you.
Thanks for having me.
Dr. Loeffler, thanks again for joining us, and we'll see you all next time.
Thank you, Microbial Insights, for having me. Thank you.